Yes.
Yeah. Okay, that's great. Thanks. So to finish off, I'm going to talk about the lipid maps, databases and tools. Many of these have actually already been covered throughout the week, which is great because it saves me talking about them, and they've been covered by people who actually are far more experts in them than I am. So this is the Lipid Maps homepage. The frequently used resources are front and center on the page. They're in this array of buttons there. So the tools and the databases that are used most frequently within the site are readily available straight from the homepage. If, however, you want more detail, then under the resources tab here at the top, pull down the menu and you get databases, tools, and a variety of other services, which I'm not going to have time to talk about this afternoon. I will mention briefly Lipid Web is a resource by Bill Christie, which has a huge amount of detailed information about lipids that he's built up over an extremely long career and almost as long a retirement working in lipids. So to start off with, on the tools section, there are 5 tabs on the tools page, of which 3 I've grayed out because they've already been talked about to some degree by people this week, which leaves me to talk about the mass spec analysis tools and the structure drawing tools, which I'll touch on very briefly, because actually I think they're all very self-explanatory. So they don't need me to talk you through them in any great detail. There's a variety of mass spec analysis tools, such as this one, which will predict the fragmentation pattern for glycosyl ceramides. So you input the ceramide head group there. So galactose, 1,3-galactose, N-acetyl galactose, neuraminic acid, galactose, glucose, acyl chain of 18 carbons, sphingosine base, 18 carbons, 1 double bond, 2 oxygens. Select the ion, select all your other parameters. And then this outputs a whole list of the fragments and the masses you would expect for those fragments. And this list goes on about 3 times the length of my screen. But you get the principle. This is what it will show you and predict how you might expect this this lipid, well, it's maybe more of a saccharide than a lipid, but how this molecule will fragment in mass spec. The structure drawing tools allow you to generate images, structures for a variety of the more common classes of lipids. And again, it's very simple. You input the parameters for this lipid. So the type of head group you have for phospholipid, the acyl chain at SN1 position, the acyl chain at the SN2 position. You press the submit button and out pops the appropriate structure, which you might use in your publications, use on your websites, presentations, wherever a lipid structure will be appropriate to use. So that's basically all I have to say about the tools, because many of them have been covered in lots more detail. So I'll go on now to talk about the Lipid Maps databases, and in particular these 4 databases that we have, not necessarily in this order. And if you click on that link I showed on the home page to databases, you arrive at this page, and up at the top the databases are named there. I'm not going to talk about the protein database. This is only a subset of UniProt anyway for particular lipid linked enzymes. So you select your database from the list at the top. And the first one I'm going to mention very briefly is the ion mobility database. And we heard lots about ion mobility yesterday. And this is essentially a list of around 1100 CCS values from the literature for lipid species. And it's a database that's suffered from maybe a lack of love recently, and there's a lot more information that we could add to that, which I'll be looking at over the next few months to see how much more, how many more CCS values we might add to it. So the results look something like this. For palmitic acid, it gives you the lipid maps identifier, the name, the mass, the species, and then the CCS data, and where we got this information from, the publication that this was derived from. And I think in all databases, that's an important piece of information to have the provenance of where the data was from. So you can go and look and critically evaluate how good you think that data is. The next databases are in silico structure database. This contains 1.1 million lipid structures generated computationally, and it's largely biology agnostic. So there's not much of a consideration about whether the acyl chains, for instance, in the glycerolipid are actually likely to occur. But it gives you a very large number of possible molecules that are chemically possible, if not biologically possible. And you can either browse the whole list or search by an individual mass overcharge ratio. So by combinatorial expansion, what I mean, in the triacyl glycerides category, there are over 700 molecules there, split initially by the sum composition, so the species level, where you have 44 carbons, 6 double bonds between the 3 acyl chains, and this is split up. The smallest we have in this database is 30, so 30 carbons between the 3 chains, no double bonds, and it goes up to 102 carbons and actually larger than that. So then you might split that, having got that sum composition, you could split that and say, well, those acyl chains might be 211 carbons and 122 carbon with 6 double bonds. And combinatorially, of course, you can have those in any of the positions. You'll note the underscores in that name, indicating, as I said on Monday, that the position of the acyl chains is not defined. But then you can drill down further to specify where the acyl chains are and where the double bonds are to get the exact structures for that. And if you search by mass, I've done this with the neutral adduct just for simplicity. Type in your mass and you'll get the results here. So it says things that fit that mass are triacyl glycerol, 44 carbons, sphingomyelins, 37 carbons, and ceramide ethanolamine. I'd forgot what that abbreviation was. Ceramide ethanolamine with 40 carbons, 4 double bonds. This gives you the sum composition or the abbreviation or the shorthand nomenclature species level, whatever we're going to call it. And I don't think we've fully arrived on a definitive name for this thing. And you can drill down further and get the species level by clicking on this link. You can get much more detail about what those lipids could be. But maybe if you don't have any more information and you don't know what this is, then really the database you should be searching is the next one I'm going to talk about, which is Compdb. So computationally generated bulk lipids. And this is another computational database using biologically commonly occurring acyl chains. And you can only search at that level. So you can only search for this species level shorthand where you don't know where the acyl chains are or where the double bonds are. It doesn't search in any greater detail. One of the most useful things about this search is you can input a very large list of precursor ion masses, should you want to. We've done it recently with about 10,000. It takes its time with 10,000, but it will get there eventually. So the procedure is you provide the list of masses, or if it's a very large list, you could upload a file into this box here. Sort the appropriate mode that you want to search for and the appropriate adduct if you have that information. Then go on to select the mass tolerance you might be looking for. And beyond that, you can search which particular classes of lipids you want to search for. If you select none of these, if you don't tick anything, it will search all of them. But you might have information, for instance, that you're not interested in the sterols, or you're only interested in sphingolipids. And the results you get back look like this. You get the name of the lipid here in just the shorthand species level identification. So N-acyl taurines, these are some of the abbreviations I know I didn't cover on Monday for brevity. Steroid, glycine conjugate, lyso-PC, etc. But these are all the things that fit that list of data. In fact, not even that list, just a single input mass could be all of these. And if lipids that match this shorthand species level ID exist in the LMSD database, then there are links through to examples there. You can also use the same interface to search the LMSD database with a similarly large list of M over Z values. So that brings me on to talk about our main database really in Lipid Maps, the Lipid Maps structure database, abbreviated to LMSD. And this contains data mainly from experimental sources. So the original Lipid Maps experiments going back a decade or more, data from the literature, and information imported from other databases such as Lipid Bank, Cyberlipids, KEBI. Metabolome is a Japanese database, maybe of plant flavonols, but all that sort of other information from other databases. And it also contains some computationally generated structures for various lipid classes. And you can see here how many lipids we have in the whole database. So for fatty acyls, 8,200 curated, so that means brought in from literature, other databases, experimental sources, and 1,800 computationally generated. And they're mostly prostaglandins and similar molecules to that have been computationally generated. Glycerolipids, you can see there are a lot computationally generated, just adding likely biological or mammalian acyl chains onto the glycerol backbone. And we're currently at 5,000, just short of 5,000, 45,700 structures. It's a Friday afternoon, I'm becoming innumerate. The search box on the Lipid Maps homepage, if you put a search term in there, then what you're searching is the LMSD, Lipid Map Structure Database that I've just talked about. So that's an immediate, quick way into the database. There are several other ways to search, and I'll briefly mention the top three of these. I'm not going to talk about programmatic access this afternoon. The first way is using the classification system, which we looked at on Monday. So you can browse the lipids, expand each of these boxes to see what lipids are in which classes. The other way is via a keyword or ontology search. So you can put in various terms into the search interface to search for particular lipids. So for instance, here, you might want to search only the fatty acyls category for a lipid where the name contains the term bromo and it has 4 or more double bonds and 1 or more rings. So you can really get quite specific in your search terms there. Believe it or not, there are 2 lipids in the database that fit those criteria. The third way you can search is actually you can draw the structure. So click on the link there and it takes you to an interface where you can sketch a molecule. So the first thing you need to do is draw the molecule. I've drawn this with various chiral centers present. Actually, it's worth pointing out that the search ignores those stereo centers. So it will find lipids with a carbon single bond there, irrespective of the chirality. And you can search by substructure, so where that molecule is part of the molecule that you're looking for, or you can toggle that box here and say, I want to search for something that is exactly that molecule, with the caveat that the stereochemistry is not taken into account. So these are the ways to search the LMSD database. What information do you get when you get to a particular lipid? So I'm going to go through the anatomy of Lipid Map structure database entry page, with a warning though first that we are in the process of redesigning these pages. So hopefully in the next couple of months, the look of these pages will be slightly different, although the information on them will be the same. In fact, more. We'll be adding more information, hopefully in this redesign, which will make them more useful to you. So the top of any molecule page at the moment, though these will move, contains the download links. So you can download the mol file that defines the structure of the molecule or various other formats with the data that's on the page present. Then there's the chemical structure drawn in standardized orientation. Now, a couple of days ago, I think it was Al Merrill talked about standardized nomenclature and standardized representations when he was saying that sphingomyelin and phosphocholine actually looked similar. And if you draw those 2 molecules in a standard orientation, you can very easily see that they do look very similar. It actually makes comparison very easy if you always draw lipids in the same way. I get very confused when I see them in completely different orientations because I have to do mental gymnastics to put them back into the standard representation that I'm used to seeing them, and to curate them into the database, I need to put them into that orientation anyway. So moving further down the page, you get to a lot of information about the molecule. Firstly, the lipid maps ID, and then a series of names for the molecule, common name, systematic, and various synonyms, the exact mass and the formula, and this pull down box here gives you the option to calculate the M/Z value for a whole variety of different adducts. And there are some missing from here. We're working on adding some and making this box selective for appropriate ones for the lipid class you're looking at. Following on from that is the classification. So in this particular case, it's a fatty acyl and it's in the eicosanoid main class and it's a prostaglandin. And then there's the shorthand. This is a fatty acid with 20 carbons, 3 double bonds and 4 extra oxygens. And then there are the links to other databases and other resources, so you can see what other databases have the same information. And I will give you a warning about that at the end of the talk, but just bear in mind lots of databases have the same information doesn't mean it's right. And then at the bottom of the page, there is the computationally readable identifiers for the molecule. And it's worth pointing out that hidden on this page, but we will make it more obvious when we redesign the pages, is a link here to show lipids that have the same structure but only differ in the stereochemistry or the bond geometry. So lipids that are stereoisomers of this one or have cis-trans double bond differences only. And following on from that, there's links to spectra in MONA or the ion mobility database if that's there, lipid standards. Then there's the status of the lipid. If it says active, then that's either curated from the literature or other databases like explained earlier. If it says active, generated by computational means, then that's the computational expansion of that molecule. And then if present, there's a reference to where we got this molecule from in the literature. This isn't present in very many of them, but I'm working on back populating that, but it's very much a work in progress and it will take some time. And then finally, calculated physicochemical properties for the molecule right at the bottom of the page. Coming shortly, we're going to add more functionality. We will be adding taxonomy information, as Val said on Monday. So we'll say which species we curated the molecule from. Now that doesn't mean it's exclusive, of course, to that species. A lipid in mouse may also be found in humans and may also be found in a cabbage plant. However, it gives you some pointers as to the likelihood of you finding that molecule in your particular sample, given you hopefully know which species your particular sample is from. So for instance, there's a steroidal saponin which is entirely responsible for the flavor of licorice, or one of the molecules responsible for the flavor of licorice. You would find that in the plant from which licorice comes from. You wouldn't find that in a lettuce, and you wouldn't find that in a human unless you've been eating licorice all morning. And last year there was actually a case of someone who died from licorice poisoning because this molecule is toxic in high amounts. And it was the particular lipid that we have in the database from the licorice plant that killed him. We're also going to be adding pathways information hopefully soon, and both Caroline immediately after me and Egon might talk about pathway information. So hopefully on a particular lipid page, you'll be able to see the molecules that this particular lipid can be transformed into and which lipids are modified to create this molecule. So that hopefully will enable you to see where it sits in any biosynthetic pathway and modify your hypotheses and your data and the information you have in the light of that. It was mentioned yesterday, and I think Ed Dennis mentioned it on Monday as well, that when Lipid Maps was first set up over 10 years ago, the aim was to catalogue all the lipids. And this is one of the early papers revealing the diversity of lipids in human plasma. So sometime on, do we have all the lipids? Well, Ed suggested on Monday that there are substantially more than 45,000 lipids in the human lipidome, and we have 45,500 lipids in LMSD. Not all are human, so the answer to that question is very definitely no. We haven't found all the lipids yet. And when I say not all are human, over the course of 2020, we added lipids from 219 different species. And actually, I made this diagram in October 2020, so it's probably more than that, actually. So we very definitely don't have all the lipids which exist out there in nature as yet. which means if you publish a new lipid structure or you know of a lipid which is missing from LMSD, and Miguel sent me a whole list of them yesterday, so I'm chugging through that at the moment, adding lots more polyketides to the database. But if you know of something, get in touch and tell us, and we can add it to the database. What do we need to add something to LMSD? Well, it's a database really of fully characterized lipids. So for a phospholipid, we really need the SN positions of the acyl chains, the location and the stereochemistry of the double bonds. We can omit chiral centers, for instance, the chirality of this hydroxyl, if it's not known. Preferably, obviously, we'd like it as fully characterized as possible, but there are some things we can get away with. So that's a brief run through LMSD. I'm going to finish with some warnings that all databases and bioinformatics tools in general should be used with care. We have over 45,000 distinct lipids. They equate to only just short of 9,000 unique masses. So every mass on average has 5 lipids attached to it. And over the course of this week, we've heard time and time again that you need to have more information than a mass to characterize a lipid. And hopefully you all know that, and that's a message you've very definitely gone home with. There are many people out there who haven't heard that message. I was looking at 2 papers only yesterday when they literally got just a mass and had assigned it to a lipid in LMSD based on that and only that. If you're one of those people, I'm prepared to sell you this gold bar that weighs 1 kilogram for half the retail price. It will come up about like that, but they're the same mass, so they must be the same thing. And finally, scientists are human. You make mistakes. We all make mistakes. You may have characterized a lipid and find out that actually you've made a mistake and what you thought you had is not what you had. Curators are also human. I make mistakes too in putting lipids into the database. I can be insufficiently caffeinated, and I'm insufficiently caffeinated at the moment, which is why I have this mug in front of me. You know, mistakes happen. If you find them, point them out. Let us know. And also, we have all those links to other databases. Databases share data. This isn't true just in lipids. This is true in protein databases and genome databases. Information is shared between all of us. And that means we also share errors. So it's easy to think, we found this molecule, it occurs in 6 of the main different databases and they all agree, so it must be right. But actually behind the scenes, they all agree because we're all swapping the same information between us. So to conclude, I can see that there are lots of comments in chat, but I've not looked at them, so we might have lots of questions. I've got another 10 minutes. No bioinformatic resources gives you the answers. They're there to generate hypotheses for you to test. You need to check your results are sensible and test them. And mostly it's the role of a scientist to be skeptical. So I'll finish there. Thank you very much for your attention. And I'm happy to try and answer some questions.